Hi everyone, welcome again to the course of structural biology. We are continuing with structural biology techniques. I have discussed about high resolution technique like X-ray, NMR. In this module, we are talking about cryo-electron microscopy. This is the last class of this module. Here, I will talk about protein data bank. I would also discuss some other databases which are relevant where structural data, structural information used to be deposited. So, today's topic is 3D structure of biological macromolecules, their databases of structures of biological macromolecule and understanding .pdb files. So, first we start with protein data bank. The protein data bank is a database for the 3-dimensional structural data of large biological molecules, or as we say biological macromolecules. The data typically obtained by X-ray crystallography, NMR spectroscopy, or as now it is coming cryo-electron microscopy and submitted by structural biologists from around the world are freely accessible on the internet. Any experimental information regarding biological macromolecules, their structural data have to be submitted and validated first before scientific publication. So, you cannot go for a scientific publication unless your data is deposited, but deposition is not enough. After deposition, people from PDB they will give you a validation certificate. So, when you show this validation certificate, then only the journals will start reviewing your paper. Currently, this is considered as the only repository for 3D biological macromolecular structure solved by using experimental technique. Anywhere, any structural information obtained through experiment have to be deposited in protein data banks. That is why it is very important database. This is the home page of PDB RCSB Research Collaboratory Structural Bioinformatics. I have gone through different courses, but just for this class I actually collect and delivered this class at the last moment, so that I could provide you the most recent data regarding the structural information. If you look at 1,75,282 biological macromolecular structure entries are deposited currently in the protein data bank. So, as I told, beside PDB there are other databases. One of them is electron microscopy database. As we are talking about cryo-electron microscopy, just now we have finished about cryo-electron microscopy. The electron microscopy data is deposited in electron microscopy database or EM data resource. Cryo-electron microscopy enables the determination of 3D structure of macromolecular complexes and cells, biological organelles from 2 to 100 angstrom resolution. We have talked about them in details. EM data resource is the unified global portal for one-step deposition and retrieval of 3D electron microscopy density maps, atomic models and associated metadata. It is a joint effort among Stanford SLAC Cryo-EM facility and the Research Collaboratory for Structural Bioinformatics, which is RCSB, in collaboration with the EMDB, the electron microscopy database team at the European Bioinformatics Institute or EBI. The major goal of EM data resource is to work with the 3D electron microscopy community to establish data validation method that can be used in the process of structure determination. as we have discussed before. So, once the data is prepared, the data have to be deposited and then the data have to be validated. Define the key indicator of a well determined structure that should accompany every deposition. So, the comparison and getting the quality of the structure. Implement appropriate validation procedures for maps and map derived models into a 3D electron microscopy validation pipeline. As I talk about, you have to validate properly. Currently, this year 3rd March, there are 14,303 EMDB map entries, 7083 PDB coordinate entries. And if you want to know more, the link is given. You go to the link and you will get the details about the electron microscopy database. Another related database is Nucleic Acid Database or NDB. This is the logo. So, a portal for 3-dimensional structural information about nucleic acids. The NDB or Nucleic Acid Database, founded in 1992 by Helen M. Berman from Rutgers University, Wilma K. Olson again from Rutgers University, and David Beveridge from Wesleyan University. The NDB contains information about experimentally determined nucleic acid and complex assemblies. So, nucleic acid small molecule, nucleic acid protein. People could use the NDB to perform searches based on annotation relating to sequence, structure and function, and to download, analyze and learn about nucleic acids. The NDB project is funded by the National Institute of Health NIH and has been funded by National Science Foundation NSF and the Department of Energy DOE in the past. Again, 3rd March 2021, number of release structure, the nucleic acid complex with small molecule with protein is 1, 1, 2, 3, 4, 11,234. Again, about further details, the link is provided here. So, interested people could go through and know more about. Biological Magnetic Resonance Data Bank or BMRB, a repository for data from NMR spectroscopy on proteins, peptides, nucleic acids and other biomolecules. So, as now it is known to us, there are 3 different techniques: X-ray crystallography, NMR and cryo-EM. So, PDB, as it is now understood by you, but I will go to history and talk about in details. PDB started with X-ray crystallography. So, it is called PDB, protein data bank, but it have protein, DNA, RNA, DNA RNA complex and virus and everything. The concept of a biomolecular NMR data bank was developed under a 5-year research grant awarded to the University of Wisconsin-Madison from the National Library of Medicine, National Institute of Health NIH. BMRB or Biological Magnetic Resonance Data Bank at the University of Wisconsin-Madison has been supported since 1st September 1996 by the National Library of Medicine. BMRB collect, annotate, archive and disseminates worldwide in the public domain the important spectral and quantitative data derived from NMR spectroscopic investigations of biologically relevant molecules. The goal of this data bank is to empower scientists in their analysis of the structure, dynamics and chemistry of biological systems and to support further development in the field of biomolecular NMR spectroscopy. So, this data bank is mostly specific towards NMR spectroscopy. Again, the link is provided so that you could go to the further details. As I told, coming to the history of protein data bank, there are 2 major forces which converge to initiate the protein data bank: a small but growing collection of sets of protein structure data determined by X-ray diffraction. I have talked about these. If you remember the history I talked in time of X-ray crystallography, the history I talked when I talk about protein structure determination, DNA structure determination, all of them started at the end of 1960s and early 1970s. Those data needed to be kept somewhere, and that was one of the force to bring the idea about protein data bank, but this was also supported by newly available molecular graphics display, the Brookhaven raster display or BRAD, to visualize these protein structures in 3D. As new structure was coming, more and more scientists want to do research on them, compare them. So, visualization was must, and that time this development of Brookhaven raster display happened, and now people want a platform. Idea of that platform initiate the idea of PDB. In 1969, with the sponsorship of Walter Hamilton at the Brookhaven National Laboratory BNL, Edgar Meyer from Texas A&M University began to write software to store atomic coordinate files in a common format to make them available for geometric and graphical evaluation. By 1971, one of Meyer's program called SEARCH enabled researchers to remotely access information from the database to study protein structure offline. This is the first time people start getting all the data and the related software to analyze them, making protein crystallography and structural biology increasing its popularity. SEARCH, the software, was instrumental in enabling networking, thus marking the functional beginning of the PDB. As it is founded officially in 1971 by Brookhaven National Laboratory, New York, it established at Brookhaven National Laboratory with collaboration of Cambridge Crystallographic Data Center from United Kingdom. In 1973, the early structures, they are here: myoglobin, hemoglobin, as we know they are the first two structures, then rubredoxin, cytochrome B5, trypsin inhibitor, chymotrypsin, carboxypeptidase, papain, certillizin, lactate dehydrogenase, all the early structures came. So, the deposited structure include all those I talked about. So, they came, and then Hamilton, who was the initiator, upon Hamilton's death in 1973, Tom Cosley took over direction of the PDB for the subsequent 20 long years. In 1974, first tRNA structure appeared. 1974, structure of East phenylalanine tRNA at 3 angstrom resolution published in Nature. In 1973, three-dimensional structure of East phenylalanine transfer RNA, folding of the polynucleotide chain published in Science. In 1979, first zDNA published, the molecular structure of a left-handed double helical DNA fragment at atomic resolution, again published in Nature. In 1981, first bDNA published. In 1981, structure of a bDNA dodecamer, conformation and dynamics proceeding of the National Academy of Sciences of the United States of America. 1982, Nobel Prize in Chemistry awarded to Aaron Klug. The prize was awarded for his development of crystallographic electron microscopy and his structural elucidation of biologically important nucleic acid protein complexes. In 1988, Nobel Prize in Chemistry again for 3D structure of photosynthetic center. A lot of structural biologists got Nobel Prize, we have discussed. So, these are marked for PDB because each of these awards actually help PDB to grow more. The Nobel Prize was awarded to Johann Dissenhofer, Robert Huber and Hartmut Missel for the determination of the three-dimensional structure of a photosynthetic reaction center. This is very critical because we understand the mechanism of photosynthesis by looking at the high resolution structure of the photosynthetic reaction center. In 1989, the first NMR structure released in PDB. So, the first NMR structure was released in PDB with PDB ID 1BDS, which belongs to the anti-hypertensive and antiviral protein BDS I or 1 from the C anemone Anemonia sulcata. Also in 1989, IUCR, International Union of Crystallographic Research, have made fixed policies, because before that people have developed PDB, but they did not think that PDB will come so far. So, now at this 1989, when the number of structure was increasing rapidly, International Union have make policy on data deposition, and they also publish the data. In 1991, the first 3D electron microscopy data, the first 3D electron microscopy structure of bacteriorhodopsin. This is one of the innovation which made the mark of electron microscopy towards solving protein structure by Henderson, who is also part of the Nobel Prize at 2017. So, in 1991, they come up with the structure of bacteriorhodopsin using cryo-electron microscopy. We have already discussed that event, but now it is also connected in terms of PDB. In 1997, Nobel Prize for mechanism of adenosine synthesis. So, the Nobel Prize was awarded in chemistry to Paul D. Boyer and John E. Walker for their elucidation of the enzymatic mechanism underlying the synthesis of adenosine. It was again a very critical step, because this is the first elucidated mechanism about the nucleic acids. 1998, inclusion of other databases. So, as I told, we talk about different databases, but now we have this WWPDB, the Global PDB Foundation, and all the related databases are working together. So, in 1998, that was started, the inclusion of other databases. Database at European Bioinformatics Institute, the EBI, it become PDB-E or PDB Europe. So, this is the logo of PDB-E protein data bank in Europe. It is transferred to the Research Collaboratory for Structural Bioinformatics or RCSB. So, one PDB-E created, and PDB now make RCSB PDB. In 1999, 10,000 structure is released from PDB. In 2000, the first ribosome structure in the PDB, and Osaka University opens a PDB data deposition center in Japan called PDBJ. 2000, a Nobel Prize again in chemistry, Kurt Uttrich was awarded with Nobel Prize for his development of nuclear magnetic resonance spectroscopy for determining the three-dimensional structure of biological macromolecules in solution. We have talked about that when we are going through NMR spectroscopy. It is a very critical event in the world of structural biology. EMDB established the electron microscopy database, which we talked about, established at MSD EBI, the European Bioinformatics Institute. 2003, it is a very important year for PDB. WWPDB established by RCSB PDB, PDB-E and PDB-J. So, all the three PDBs, they merged the PDB, PDB Europe and PDB Japan, they merged together to form the global PDB. This is the logo Worldwide Protein Data Bank, WWPDB. In 2006, Nobel Prize in Chemistry to Roger D. Kornberg for his studies of the molecular basis of eukaryotic transcription, another landmark discovery in structural biology. BMRB, which we talked at the beginning, the Biological Magnetic Resonance Bank, joined the WWPDB. In 2008, 50,000 structure is released, and if another very significant thing happened, now it became mandatory to deposit the X-ray and NMR data if you want to publish. Before people try to escape and all, now it become mandatory. In 2009, Nobel Prize in Chemistry, Venkat Raman Ramakrishnan, Thomas S. Teeds and Ada Yanath. So, Venki and Thomas from US and Ada Yanath from Israel for studies of the structure and function of the ribosome. 2010, Worldwide Protein Databank Foundation established. So, now they make Worldwide Protein Databank Foundation. WWPDB was there, it is Worldwide Protein Databank Foundation. Deposition of chemical shift data mandatory for NMR data. So, if you remember, when we are going through NMR spectroscopy, we talk about the chemical shift, how important it is, because you have the spectras and the spectra is influenced by the chemical environment, that is called chemical shift. In 2011, first structure from an X-ray free electron laser by Chapman et al. 2012, again Nobel Prize in Chemistry to Robert Lepkowitz and Brian K. Kobilka for studies of G protein coupled receptors. Again a very landmark discovery, because G protein coupled receptors are already known to be associated with a lot of drug transport, and also a lot of disease related phenomena were associated with, but in 2012 with the advent of this high resolution structure, a lot of things was further established. In 2013, now structural biology is definitely still in the leading role, but a lot of computational initiatives are grown. So, a lot of people who did not take care of some part of the crystals or some part of the structure when they are working with the structure, because once they get what they want to get, they just publish the, I would say it is not wrong, but with small mistakes and all, they did not take care, but those mistakes become really critical for the computer scientists to calculate things. And now in 2013, with a thorough meeting between the structural biologists and computational biologists around the world, they make the validation report mandatory. What is the validation report? The validation report is a report like before, when you submit the data, you submit it, if you maintain the rough formats, they will allow you to submit, and within few days you get, or probably whenever you submit you get a PDB ID. Now, with this PDB ID you could continue. Now, it is not. You submit the data, if you are a crystallographer, another crystallographer like you who is now a staff of the WWPDB will check your data, do the refinement by their own, and after being ensured they will provide a good validation report. Otherwise, they would communicate with you, they will ask you to make changes, you will do the changes, and then the validation report you will get. And as I told, only after getting this validation report you are allowed to go for scientific publication. So, the validation report required for manuscript submission to Journal of Biological Chemistry or anything, that makes a huge improvement in the quality of data. So, it is a very significant step, especially considering computational biology. At the same time, see how coincident, at the same time the year when initiatives are made to help computational biologists, the same year Nobel Prize in Chemistry was awarded to Martin Karplus, Michael Levitt and Area Wursel. It called that for the development of multi-scale models for complex chemical systems, but actually as a structural and computational biology, as a part of this community, we all believe that this Nobel Prize was awarded to a field to acknowledge its contribution to the revolution of biology research. It is given to the field of dynamic study. Biology could never be understood perfectly at the static level. So, incorporation of dynamics is a must, but because of the enormous volume and size of this biologically complex systems, it takes a huge time, but not now. With the contribution of many, but I would just talk about Martin Karplus, who have collected all the programs and written a program package in C language, C++, which is the part of the program package CHUM, the globalization of computational biology was started. And this Nobel Prize is the acknowledgement of that. In 2014, integration of large structures with the main PDB archive using PDB-X MMZip, previously split across multiple entries in traditional PDB file format. So, now you get them together with the introduction of MMZip. Now, there is also option of MMTip. Now, greater than 1 lakh structures in the archive. Another very significant step, introduction of OneDep, a one or global deposition system, which is called a new system for deposition, validation, curation. It become user friendly, it become automatically validated. So, it improves the quality of the structure. Before deposition, you know that whatever mistakes you have made, so you have corrected them, so the things speed up. UNESCO celebrates the year of 2014, the International Year of Crystallography. In 2016, the deposition of electron microscopy maps mandatory. If you remember, the year of 2016 was considered cryo-electron microscopy as a technique of the year, followed by the Nobel Prize to Dubochet, Henderson, as we have already discussed, and Joachim Frank. In 2016, new validation report provided for NMR and 3-dimensional electron microscopy. In 2017, as I was talking about, Nobel Prize in Chemistry to Jack Dubochet, Joachim Frank, Richard Henderson for developing cryo-electron microscopy for the high resolution structure determination of biomolecules in solution. We have discussed this already. PDB Dev, a prototype system for integrative hybrid structural model, which is announced in structure. So, now, before there was technical improvement was going on, mostly the structure solutions was based on X-ray crystallography, NMR was helping, but now we are on the technical peak where all of them, especially cryo-EM is providing you a very high resolution. So, hybrid methods start coming. In 2018, Nobel Prize awarded to Francis H. Arnold for the directed evolution of enzyme, or you could say enzyme engineering. So, enzyme engineering is a very modern technology with modern demands, and this is only possible by utilizing high resolution structures. So, that is why it is a part of PDB. In 2019, there is improved ligand validation, greater than 1,50,000 structure entries in the PDB archive. 2020, Nobel Prize in Chemistry, Emmanuel Charpentier and Jennifer A. Doudna for the development of a method of genome editing, the CRISPR technology, which actually revolutionized the world of molecular biology, the target based modifications, and the Nobel Prize is awarded to the deserved candidates. Again, the targeting structure have a huge role in there. In the same year, Nobel Prize in Physiology Medicine to Harvey Alter, Michael Houghton and Charles M. Rice for the discovery of hepatitis C virus in a year where most of the scientific activity was stopped because another virus, the COVID-19. So, first SARS-CoV-2 structure in the PDB, which is COVID-19 main protease. Validation reports for 3D electron microscopy model coordinates and maps the more improvements of creatine microscopy, improved carbohydrate data and representation released, and 2021, this year where we are standing, it is PDB 50, the golden anniversary of the protein data bank. Starting in 1971, it now touched in 2021, it touched the 50 years of its journey. What is the vision of PDB? Sustain freely accessible interoperating core archives of structured data and metadata for biological macromolecules as an enduring public good to promote basic and applied research and education across the science. What are the missions of PDB? Manage the WWPDB core archive as a public good according to the FAIR principles, provide expert deposition, validation, biocuration and remediation services at no charge to data depositors worldwide. This is an amazing service which is helping the structural biology to keep the standard improved, keep working on them and easy to deposit, validate, so that now or still now we have one platform for all the structures. Ensure universal open access to public domain structural biology data with no limitation on usage. For this reason, we could access to any structure at any moment at free of cost. This is a great service. Develop and promote community endorsed data standards for archiving and exchange of global structural biology data. So, if you look at the functional component how PDB work, they could basically divide into 6 parts: deposit, search, visualize, analyze, download and learn. Deposition, you could understand, you have a new data you want to deposit. Deposit have 3 stages: prepare, validate and deposit. For data extraction, PDB underscore extract and harvest data in PDB XMMC format from structure determination programs when it is done. SF tool, I have discussed about them at the end of the X-ray crystallography module when I was showing you how to deposit. So, again we deposited in the PDB, so I am repeating this part. SF tool convert the structure factor files among various formats. So, these are for large biological macromolecules or biological molecules. For small molecule, you have ligand expo, it search in chemical component dictionary, the CCD for the IDs of the released ligands. Data format conversion, PDB ML to CIF, convert PDB ML format data into PDBX MMC format. Point suit generate symmetry records for macromolecular assemblies with point and helical symmetries. Maxit translate data between file formats and more. Validation, as I told you, need a validation report. The validation report contain an assessment of the quality of a structure and highlight specific concern by considering the coordinates of the model, the experimental data and the fit between the two. So, it needed to be fit between your experimental data and the model you have developed. Easily interpretable summary information that compares the quality of a model with that of other models in the archive will help users of PDB data to critically assess archive entries and to select the most appropriate structural models for their needs. So, as I told, there is a automation now. We have already established structures, their qualities are known. So, once you would put your data, the data would be compared with the existing structural data in the database and they will tell you about what you need to work on or not, or what is the quality. These reports are developed using the recommendation of the WWPDB validation task forces. As I told, you are a crystallographer, you are a NMR specialist, you are a cryo-EM scientist. Like you are working in your lab, you are writing funds and you are getting money, your students are working, you get a new structure solved, but there are a group of people who have the similar training like yours. They are continuously taking care of the deposited data, so that the quality would not be down. Reports for released entries are available from structure summary pages. So, you could understand, you could get the recent structures and all. Validation reports for manuscript reviewers are created during the annotation of deposited structures. So, that is the whole process. Then, as I told, the deposition is in one depth system. This is the logo of one depth. You have to log in and you have to go there and you have to deposit stepwise. So, because of time lag, I am not going into details of the deposition. Given opportunity, I could come up and talk in a different course, but now even if I am not talking about, it would not that I am not giving you the opportunity to learn. So, here is a very nice video which will show you how to deposit the coordinate, how to do the NMR specific deposition, how to do electron microscopy volume map deposition. If you follow them, it would be very easy to make the deposition. Coming to search, PDB, the protein data bank, have made a lot of different options for searching: basic search, advanced search, sequence search, unreleased and new entries, browse by annotation, explore the archive, PDB statistics, ligand, drug targets. You have to change the words and do the searches and you will get what you want to get. Viewers means the platform to make the view. So, one is 3D structure viewer, like you could see here, there is a PDB ID 4HHB showing a X-ray crystal structure, 3J3Q showing a cryo-electron structure and 1 BTN as a NMR structure. So, to visualize them you need a platform. I am not talking much here because in the next module, an entire module, we are talking about the visualization, calculation, structure factors and all these things. There are sequence viewers also. One is protein feature view. It provides a graphical summary of biological and structural protein features of PDB entities and how they correspond to the UniProt KB sequences. If you remember, when I was introducing databases, I have introduced two databases. One is protein data bank, which we are spending an entire class on that. Another is UniProt. So, protein data bank have the information about the 3D structure, UniProt have the information about the sequence, and you could correlate them with the protein feature view. It loads feature from RCSB and third party resources such as UniProt KB as I talked about, CAT or Scopy, the other databases. Another is genome view. It illustrates the correspondences between PDB entity sequences and genomes. When the relationship between PDB entities and genes are available, PDB entity sequence are mapped to their genome positions to show which region of a gene are available in the entity coordinates. Download, you could download coordinate and experimental data files. What you have to do? You have to enter the PDB ID, which I am talking continuously. PDB ID is combination of number and letters like 2GDN is a PDB ID. You have to remember there are 4 positions. The first position have to be a number. So, PDB ID is a unique ID for a structure. In the PDB ID, there are 4 combination of 4 number and letters. Numbers could be anywhere, letters could be only in the 3 position. This number have significance, higher the number, more recent is the structure. Coordinate data files can be downloaded in the following formats: PDB, PDB-X MMZip, PDB-ML XML, PDB-ML XML with the header, biological assemblies in PDB, biological assemblies in PDB-X MMZip. Experimental data files can be downloaded in the following formats: structure factors, NMR restraints, chemical shifts and NMR restraint, a new version. Learn, protein data bank have contributed significantly towards the learning of common students regarding the structural development and how those biological macromolecular structure have contributed to biology. David Goodsell is a name who is continuously writing about molecule of the month. Molecule of the month is a learning session of PDB where the best or potentially most important structures solved in a current month which have immense biological importance, David Goodsell write about that. It is also working as an educational resource. It provides material that promote exploration in the world of protein and nucleic acids. It have curricula, authentic hands on teaching material, individual and group activities. A lot of teachers could get benefited from those materials. News and events, upcoming meetings and events which RCSB is holding. Guide to PDB data, understanding PDB data is a reference to help explore and interpret individual PDB entries. And another very interesting thing is G's Digital Archive. It is a view of iconic illustration by the gifted artist Irving Jace in context with PDB structures and educational information. You will see a lot of illustrations which are really easy to understand, but scientifically enriched. So, these are some of the facilities which we get in PDB. Now, I will talk about the growth of PDB. As you see, starting in 1970, now it is growing, and if you look at number, as I have already told, the number is 1,75,282 total entry, and each year the entry is increasing. Currently, in the last year 2020, we have 14,000 new entries. So, there is a steady growth observed in the deposition of different structural entries. 2000 to 2010, a marked increase is observed. This is because of the introduction of structural genomics, which I talked in the X-ray crystallography part. Structural genomics comes up with lot of potential. They have delivered a lot of structures, but they could not have lifted the up to the expectation which next generation sequencing have provided us. So, gradually the projects are being closed. Currently there exist 1,75,282 entries. How much PDB is viewed this protein data bank? If you see, this is the total numbers each year starting from 2009. This is the FTP archive and this is the total website. So, as I told, the question is how many people use the data deposited to protein data bank? Straightforward answer in 2019, 84 crores the time it was viewed the protein data bank. Website, you know, directly you go to the website, the FTP, which is a file transfer protocol, is the way that files are moved from one location to another on the internet. So, people when they have downloaded structure and all, this comes here, this is 51 crore and through website around 32.6 crores. It is a huge number. As I told, there is a marked increase. You see in 1992 192, 696, 1289, it is reduced little bit, but then steady increase. So, this is the major time of structural genomics. This is very important. This is the current holding breakdown at the protein data bank. If you look at that, X-ray 1,36,983 protein only, NMR 11,613 only protein, electron microscopy 4,725. There are other methods, neutron diffraction, combined methods and all, but if you combine protein oligosaccharide for X-ray 8,000, electron microscopy 655, whereas NMR 31, protein nucleic acid again 7226 for X-ray, 270 for NMR and 1632 for electron microscopy, showing the steady increase of a potential technique which we discussed in our previous classes. So, total through X-ray 1,54,628 structures, through NMR 13,298, electron microscopy 7,000, but if you remember we have shown in 2017 to 18 EM have already started superseding NMR. So, and the total is 1,75,282, we have already talked about that. PDB also encourage undergrad and young people towards development of different type of modeling material. They arrange competitions which actually help seeding the concept of structural biology in the young minds. We are talking mainly about X-ray crystallography solution, NMR and cryo-EM, but the PDB entries are not limited to these three techniques. It is also contributed by neutron diffraction, electron crystallography, solid state NMR, solution scattering, fiber diffraction, powder diffraction, EPR, infrared and fluorescence transfer. So, there are number of methods which contributes to PDB. As we all know X-ray clearly dominates, which is followed by solution state NMR, but cryo EM is upcoming with amazing potential. So, you could search the PDB file by typing the PDB ID directly, you search and you get the PDB ID. Now, how you would work on them, how you would download it, how you would do the analysis, we will talk about them, and in the visualization and understanding PDB section in the next module, where I will show you what are the components of the PDB file, how the coordinates are kept, what are the different things other than coordinates in the PDB file, and how the PDB files of X-ray, NMR and cryo-electron microscopy differs, and all of this. Thank you very much, you guys being an amazing listener. Please keep following us, listening to the classes, and send your questions if you have queries. Thank you very much.